Delighted to introduce Matthew Porteus, a professor at Stanford University at the School of Medicine.
Hello, and thank you for the invitation to present at GeneScript's Gene and Cell Engineering Virtual Summit in 2021. This virtual world is something that is interesting, but hopefully we'll get back to in-person meetings in the near future. Today I will be talking about targeted integration in stem cells, and the subtitle is Generating Genetically Engineered Cellular Drugs. I noticed that there are two tracks, and concurrent to my talk is a talk by Chandra Pruitt-Miller from St. Jude's, who's talking about I can't even say it. Anyway, genome editing across a broad range of genomic and cellular targets, and I really advise you to listen to her. She has tremendous practical knowledge about how to use genome editing. So I do have some potential conflicts of interest, and I will, including equity and serving as on advisory boards for several different private companies. I think the history of medicine has demonstrated that with new technologies comes the ability to improve the health and lives of patients throughout the world. And sanitation and clean water still is the #1 way to improve the quality of life of people around the world, affecting tens and if not hundreds of millions of people or billions of people. Nonetheless, with the development of antisepsis and anesthesia, surgery became a viable option to treat patients. We are reminded this year about the importance of vaccines in contributing to human health and our ability of arming our immune system to prevent devastating infections. The bread and butter, I think most people think in medicine, are small molecule drugs, but in the last 30 years, biologics, so enzymes and antibodies, are now almost as much the bread and butter of medicine as small molecule drugs. But I see in the next 20 to 30 years that cell and gene medicines, living drugs, I hope will one day become as integral to medicine and the improvement of lives of patients in the U.S. and around the world as all these other prior modalities. An example of what I mean by a living drug is shown in this experiment. So we took engineered, and I'll talk to you how we do this, genetically engineered human hematopoietic stem cells, and we transplanted them into a mouse. These stem cells were labeled with a luciferase gene, so it allowed us to do in vivo tracking. And at time 0, you can see the cells distributed throughout the body, and then 3 days later, they localized to the site of the injection at in the femur. And you can see over time that these cells expanded and migrated to the different bones of the body, including to the cranium, and different organs of the body, including the liver and spleen, such that by 4 months post-injection, our drug now had expanded and migrated to different parts of the body, which is very different than other types of drugs, and is why I think people are calling these types of drugs living drugs. So we're very interested in using the precision of targeted integration to engineer these cellular drugs. And this takes advantage of the genome editing pathway. We use nuclease-based genome editing, in which we design a nuclease to make a specific break. But then instead of allowing the break to repair in a mutagenic fashion, we harness the homologous recombination pathway so that we can make either single base pair changes or target the insertions of full genes into the site of the break. Now, this idea of using homologous recombination to treat diseases is not a new one. And I cite this paper from Dr. Oliver Smithies, who passed away several years ago, who demonstrated in 1985 that he could target the beta globin locus in a human cancer cell line. The frequency was extremely low, but this proof of concept that one could do targeted integration is what my lab has been building on for the last couple decades. I'll note that Oliver won the Nobel Prize for developing homologous recombination based gene targeting in mouse embryonic stem cells. So there are a number of different ways one can use homologous recombination to modify the genome, and I outlined 4 here. They include the change of just a single base, the insertion of a gene back into itself, so-called functional gene correction, the insertion of a promoter and gene into a safe harbor, a genomic safe harbor, such that every cell has the same integration site and the same cassette. And finally, the integration of one gene into another gene, such that you capture or you repurpose the endogenous regulatory elements to express a second gene. And over the next 30 minutes or so, I will review how we've applied homologous recombination in each of these ways. The system we use is an ex vivo system in which we purify our cells of interest. We put them into cycle because the recombination process is most active in S and G2 of the cell cycle. We deliver our CRISPR Cas9 nuclease as a ribonucleoprotein complex, complexing a high fidelity Cas9 protein developed by IDT to a synthetic guide molecule that has end modifications to prevent its degradation. And we deliver this RNP complex via electroporation. We then deliver our donor template using a non-integrating viral vector. The reason we use a non-integrating viral vector, adeno-associated virus, in particular serotype 6, is it has evolved mechanisms to deliver its DNA content to the nucleus of cells while mostly avoiding the cellular sensors in the cytoplasm to unnatural nucleic acids or foreign DNA. And this system is highly effective across a broad range of different primary human cell types shown on the right-hand side. But today I will be focusing on applying this system in human hematopoietic stem and progenitor cells. So the first example I'm going to give is of using homologous recombination to make a single nucleotide change. We have been working on applying this for sickle cell disease for a number of years. We're fortunate in 2021 to be able to publish our IND-enabling work in Science Translational Medicine, led by Annalisa Latanzi, Joe Camarena, Danny Dever, and many others. I have highlighted Dr. Nihar Bhatia's name because unfortunately, while she was essential in this work, she passed away just before the work was published. So we honor her and her legacy and her efforts through this. And in this work, what we're going to be doing is genetically correcting a missing function in the gene. And I'll talk about other examples of how we can use homologous recombination to add a new therapeutic function, so-called synthetic biology. And we've applied this to, as I mentioned, a disease called sickle cell disease. Sickle cell disease is probably the most common serious genetic disease in the world. It is caused by a single nucleotide mutation or variant in the human beta globin gene, in which an A is mutated to a T, causing a single amino acid chain. Glutamic acid is changed to a valine. And this results in the polymerization of the hemoglobin molecule under low oxygen conditions. And the hemoglobin molecule is only expressed in red blood cells. And when the hemoglobin tetramer polymerizes, it converts the round red blood cells into sickle-shaped red blood cells, giving the disease its name. In Africa, where millions of people where 300,000 people, approximately 300,000 people are born per year with sickle cell disease, the median lifespan, unfortunately, remains in the first decade of life. In the United States and other in Western European countries, the median lifespan for this disease is in the mid-40s, although patients with sickle cell disease have multiple manifestations of the disease, including frequent painful bony crisis, shown in the painting on the lower left, as well as neurocognitive problems and end organ destruction. In 1984, a proof of concept that one could cure sickle cell disease using genetically corrected hematopoietic stem cells was performed at St. Jude's Children's Hospital. And this was a young girl who had cancer, as well as a cancer called AML, as well as sickle cell disease. And she received a bone marrow transplant from her brother, immune-matched sibling donor. and her brother had sickle cell trait. And 15 months after the transplant, her red blood cell system adopted the characteristics of her donor, of her brother, and she no longer had sickle cell disease. And not only was she cured then of her sickle cell disease, she was also cured of her AML. And now, 30 years later, she remains alive, or over 30 years later, she remains alive and well from this process. So this proved that genetically corrected cells could cure the disease. But the problem is that most patients with sickle cell disease do not have an immune-matched donor. And even in allogeneic bone marrow transplantation, there are significant immune problems that are part of the transplant process that can cause problems. So the idea is to mimic what happens in an allogeneic bone marrow transplant by using gene editing to correct the mutation in the patient's own cells. In this case, every patient could be their own donor. And work that, again, was led by many different people, including Annalisa Latanzi and Rasmus Bach and Joe Camarena and Danny Dever. We showed in 2016 that we could correct at the allele, we could get 60 to 70% allele correction in CD34 cells derived from 6 different sickle cell disease patients using a donor vector designed to correct the mutation, to inactivate the cutting of the allele after correction occurred by making small changes in the PAM and the guide binding site, as well as creating small changes, silent changes between the cut site and the change we wanted to make, highlighting the importance of thinking carefully and precisely about the donor design as you want to harness the homologous recombination process. When we converted these hematopoietic stem and progenitor cells into red blood cells in vitro, we found that essentially all of the pathologic hemoglobin S was converted to the non-pathologic hemoglobin A. Moreover, we had evidence that the long-term hematopoietic stem cell, here labeled HSC, was being modified at high frequencies. So in work done by Ron Bake, he was able to show that in the heterogeneous CD34 positive bulk population, the correction frequency ranged between 70 and 80% in 4 different donors of CD34 cells, but in the phenotypic long-term HSC, it ranged between 50 and 70%, a slight decrease, but still a significant number. And following transplantation of these cells into mice, into immunodeficient mice, we find over on the right-hand side that the engraftment of the gene-corrected cells using CD34 cells from sickle cell disease donors exceeds the clinical threshold that is thought to be needed to achieve a cure of this disease. There is some discussion of whether the threshold is 5%, what I call the Walters line, or 15 to 20%, what I call the Tisdale or Fitzhugh line. Nonetheless, we see that Plerexa4 mobilized CD34 cells from sickle cell disease patients can be genetically corrected and engrafted into an immunodeficient mice with preservation of a large percentage of the gene-corrected cells. The human into mouse model has its strengths and weaknesses. So in collaboration with Adam Wilkinson and Hiro Nakauchi, my lab neighbors here at Stanford, we took advantage of the Towns mouse model of sickle cell disease, where we used the exact same editing reagents that had been designed for the human gene, and were able to modify the mouse HSCs because Tim Towns and his colleagues had replaced the human genes with the mouse genes. The first thing Adam and the team were able to show is that in contrast to the human into mouse experiments, we found that the mouse into mouse experiments, we would get approximately 25% targeted integration in the mouse HSC, and following transplantation, that percentage of targeted integration was maintained. We did not see a drop, which supports the idea that at least part of the reason we would see a drop of human-corrected HSCs into the mouse model had to do with the xenogenic nature of the model. So we could directly use this model to see if we could correct the mouse disease, and we found that the overall correction frequency in mouse HSCs was lower, approximately 20%, because we had not optimized the system for mouse HSCs. I remind you, we were getting 60 to 80% in human HSCs. But following transplantation of the mouse HSCs into the Townes mouse model, we could show that in a large fraction of the mice, we could generate percentages of hemoglobin A that were curative of the mice. So more preclinical support that this might work in humans. These experiments were all done at laboratory scale, and so we then, in collaboration with our colleagues in our GMP facility, we were able to show we could scale this process up to clinical scale, where we can modify, instead of 1 or 2 million cells at a time, hundreds of millions of cells at a time, and this could be done in a highly reproducible fashion. So this program has been licensed to Graphite Bio, who have licensed the IND and are working towards initiating a first-in-human phase 1/2 clinical trial for gene correction for sickle cell disease. And we hope to be enrolling our first patients and treating our first patients in 2021. The second application of homologous recombination is to knock a gene back into its gene. And I'm going to use a disease called SCIDX1, or bubble boy disease, as an example of why this might be a very powerful way to genetically correct disease-causing mutations. So again, we're going to correct a missing function in the cells. So SCIDX1, unlike sickle cell disease, is caused by mutations throughout a gene. In this case, the gene is the receptor for interleukin-2, called the interleukin-2 receptor gamma gamma chain gene, or sometimes called the gamma gene. And these mutations that cause the disease are scattered throughout the genome, are scattered throughout the gene. And so one couldn't just design one correction strategy for each single mutation. And in work led by Dr. Mara Pavel-Dinu, she developed a universal correction strategy. Now, I will point out that the advantage of knocking a gene into a gene, as I'll show you in a second, is that you preserve the endogenous regulation of that gene. And there are many, many genes in which preserving the endogenous regulation is important, both for the safety of the genetically engineered cells and the efficacy of the genetically engineered cells. And the strategy is to take a full cDNA, in which all of the exons are fused without introns, and target this so-called superexon into the start codon of the natural gene. So thereby, the natural regulatory elements would drive expression of the superexon that is wild type before any of the mutations were hit. And in this way, the corrected protein would be expressed appropriately in a lineage-specific manner. And so Mara was able to show, both in fresh cord blood cells and in mobilized CD34 cell, peripheral blood mobilized CD34 cells, she could get a range between 20 and 60% targeted integration of the cDNA into the natural locus. And in collaboration with Succeed Raven and Harry Malik, who generously provided us CD34 cells from patients with this disease, we could reproducibly get between 30 and 45% targeted integration. It's been predicted both from studies of humans and in mouse models that a 5 to 10% targeted integration frequency would be curative because of the selective advantage of the genetically corrected lymphocyte progenitors. We found that the signaling was rescued through the knock-in. So we can show that not only is the amount of the number of cells with phosphorylated STAT5, which is downstream from this receptor, the same between wild-type knock-in and significantly improved over knockout cells, but that the overall level of phosphorylated STAT5 approaches wild-type levels. One can see it's not quite wild-type levels, but it is sufficient to rescue the proliferation defect that is present in the knockout cells. Moreover, in an in vitro model of lymphocyte development using a organoid model, again in collaboration with Saxi and Harry, they were able to show that the knock-in approach was better able to rescue NK cell differentiation as compared to a lentiviral approach, showing that the regulated expression, at least within the NK lineage, was better at rescuing the development of NK cells than the semi-random integrations with a ubiquitous promoter that occurs using lentiviruses. Finally, the cells were able to engraft following serial transplantation. Again, we're using human modified cells and transplanting into immunodeficient mice, and we see a drop in the percentage of gene-corrected cells in this xenogenic manner, depending on whether they are transplanted intrahepatically or intrafemorally. But nonetheless, even following serial transplantation, a marker of modification of long-term hematopoietic stem cells, we achieved genetic correction frequencies above what is thought to be needed to cure this disease. So again, this program has now been licensed to Graphite Bio, who are finalizing the process development and regulatory to hopefully open a clinical trial for this disease in 2022. Point out that this idea of knocking a gene into its own gene has now been applied to a large number of different diseases, including diseases affecting the immune system like Wiskott-Aldrich syndrome, RAG2 deficiency, chronic granulomatous disease, CD40 ligand deficiency, and deficiency in FOXP3 causing genetic autoimmunity. It's also been applied to genetic red blood cell defects, including pyruvate kinase deficiency. And finally, Sriram Vaidyanathan in the lab has applied the similar strategy to create a universal strategy to correct essentially all mutations causing cystic fibrosis in airway basal cells. So, and this strategy has been applied broadly from a number of different labs from a number of different countries. So the third example I'd like to turn to is the idea of using homologous recombination to knock a promoter and gene into a single site into the genome, a so-called safe harbor. And in this way, we can add a new therapeutic function to the cells. We can ask the cells to do something that they don't normally do. And this was work that was led by Dr. Natalia Gomez-Espina in my lab, and now she runs her own independent lab at Stanford in the Department of Pediatric Genetics, or the Division of Pediatric Genetics, and she was ably assisted by soon-to-be Dr. Sam Scherenberg. She is a MD PhD student now at Stanford, but she did this work while she was an undergraduate, working with Natalia and myself. And the idea is to use the hematopoietic system as a way of delivering missing enzymes or proteins to all tissues of the body. So the idea would be to take autologous hematopoietic stem cells, use homologous recombination to knock a gene cassette into a safe harbor, transplant these cells back into the patient or mouse, and then the progeny of the hematopoietic stem cells, particularly the macrophages, which have the biologic property of migrating to every tissue in the body, including the muscle, liver, spleen, and the brain, would then serve as a cellular delivery vehicle for a missing protein. And as I noted before, this work was published in Nature Communications, so more details can be found there. But the exciting thing was to see that not only could gene targeting be efficiently done at a safe harbor locus, up to 40 to 70% knock-in, depending on the cell type, but that when this knock-in occurred, we were generating super physiologic levels of enzyme expression, in this case iduronidase. And this, so these cells were made potent by a synthetic biology, more potent by a synthetic biology approach. And when the human cells that were expressing supraphysiologic levels of the enzyme were transplanted into a mouse model of the disease, Natalia and her team could show that there were improvements in neurobehavioral activities and, importantly, improvements in the inflammatory microgliosis that is seen in this disease. So demonstration that the delivery of a missing enzyme through the hematopoietic system from a genome-edited cell can rescue a mouse model of a human disease. Finally, now not in that example I showed, we were using a ubiquitous promoter to express the enzyme. Long-term hematopoietic stem cells are very sensitive, however, to misfolded proteins or the unfolded protein response, most ably demonstrated by Dr. John Dick and his colleagues. And so what Natalia and Sam then did, along with Dina Paletta, is show that one could, instead of using a ubiquitous promoter, as one could use a macrophage or myeloid lineage-specific promoter, knock it into a safe harbor, and achieve myeloid specific expression of your transgene, thereby sparing the HSC from having to express this unnatural protein. So this concept of using lineage-specific promoters into a safe harbor is also one that I think has a lot of potential moving forward. And in the last few minutes, I just want to move to a fourth example, which is to knock a gene back into a gene. And this was led by Kyle Cromer and Joe Camarena, and it was applied to a genetic disease related to sickle cell disease called beta thalassemia major. And it was recently published in Nature Medicine. So I'll go through that work briefly in the next few minutes. So beta thalassemia, as I mentioned, is a cousin of sickle cell disease. It's a cousin of sickle cell disease because mutations occur in the same gene, the human beta globin gene. But unlike in sickle cell disease, where every patient has the same single nucleotide mutation that causes the same amino acid change, causing a pathologic protein, in beta thalassemia, the mutations are scattered throughout the gene and cause a loss of expression of the protein. So there's no beta globin made. What this results in is in the hemoglobin tetramer, which usually contains 2 beta globin proteins and 2 alpha globin proteins, there are no beta globin proteins, and so you get alpha globin aggregates. These precipitate and cause the death of red cell precursors. So so what we have then is a problem that there is too little beta globin and too much alpha globin. And David Weatherall and Doug Higgs and many of their colleagues have actually shown that one can lessen the severity of beta thalassemia major if one could eliminate 1 or 2 of the alpha globin genes. So we thought the idea we came up with was to achieve both events with achieve both goals, increasing beta globin and decreasing alpha globin through a single genome editing event. And that would be to knock beta globin into 1 of the 2 alpha globin genes. Turns out that alpha globin has 2 copies, alpha 1 and alpha 2. And if you're missing 1 or 2 of these alpha globin genes, you are healthy and have something called alpha thalassemia trait. So that is shown schematically here, where we replace the alpha 1 with with a beta globin gene and hopefully restore this beta to alpha globin balance. So the first thing that Kyle had to do was identify guide RNAs that would cut 1 alpha globin but not the other. Our RNP system is so effective that if it cut both genes, we would generate alpha thalassemia major. So we needed to have a specific guide RNA. He identified this guide RNA 5 as one that cuts alpha globin and not beta globin. And he was able to demonstrate that he could get almost 40%, or a median of 40% replacement of the alpha globin gene with a GFP expression cassette. So swapping out the entire alpha globin gene with a GFP gene, so something that we call whole gene replacement. So of course, turning cells green is a good research tool, but we needed to show that we could do that with the beta globin gene. And so he showed, again, instead of creating a targeting construct that expressed GFP, he created a donor construct that would knock the beta globin gene into the alpha globin gene. He would replace one of the alpha 1s with the entire beta globin gene. He explored the different UTRs. He explored the structure of the donor construct in terms of the length of the homology arms, and found that when he lengthened the homology arm on the 5' end, again, he could get replacement of the alpha 1 gene with the beta gene in up to 40% of the alleles in a wide variety of healthy donor CD34 cells. So, and was able to show that the level of expression from the alpha globin knock-in was proportional to the degree of knock-in, the knock-in frequency. So the more knock-in you got along the x-axis, the higher levels of hemoglobin A, and we could measure that as a percent of beta hemoglobin because we could differentiate in sickle cell derived cells where hemoglobin S is at a different peak than beta hemoglobin. See, there was a directly proportional amount related to the knock-in frequency. So finally, we were again fortunate enough to receive CD34 cells from beta thalassemia patients that have been mobilized by GCSF from John Tisdale, and was able to show that in these patient-derived CD34 cells, we could get a very high knock-in frequency of close to 50%, and that when these cells were differentiated into red blood cells, we normalized the ratio at the mRNA level between alpha globin and beta globin. And when the protein analysis was done by Vivian Sheehan's group, we showed that the ratio of beta to alpha was 0.5. This ratio of 0.5 is what is found in beta thalassemia trait, a condition in which tens of millions of people around the world are healthy, and most of whom don't even know they have beta thalassemia trait. So that ratio reaches a level that would be healthy. So we are now, again, moving this program forward in terms of process development and scaling up and safety and toxicology studies to hopefully start a clinical trial in the near future. To sum, I am highly optimistic that genome-edited cells will be the next generation of biologic and therapeutic modality that will become available to patients around the world. I think it's the precision of genome editing not only can enhance the efficacy of cells, but also the safety of those cells. And with enhanced safety and efficacy opens the possibility that they can be used in more patients and more diseases, not just patients who have serious life-threatening genetic diseases. To conclude, I want to thank you for your attention and the opportunity to present our work at this virtual conference. I've been fortunate to work with outstanding people in my lab. The people in my lab, currently in my lab, are listed here, and I've tried to acknowledge them as they've gone through, some of whom have now moved on to their next position in their career, which is exciting for me. This work has been supported by a mixture of philanthropic grants, support from the NHLBI and the Cure Sickle Cell Initiative, from California Institute of Regenerative Medicine, and a whole host of the Chan Zuckerberg BioHub and a whole host of other entities, and I thank them. I've had fantastic collaborators in our division of stem cell transplantation and regenerative medicine, and collaborators in the Stanford GMP facility who help us move what we're doing in the lab at the clinical scale and under GMP conditions that we can one day give these patients to patients as part of our broader initiative in cell and gene therapy at Stanford that we call our Center for Definitive and Curative Medicine. And finally, I want to thank our wide range of collaborators, both in academia and industry, in the US and Europe and across the world. It is these kind of collaborations that both make our work successful, but also make our work so fulfilling. So again, thank you very much, and I look forward to taking any questions.
Great, thank you, Matthew, for the talk. We can take a few questions until you have to go in about 5 minutes. So there's a question here from Ray. What are your opinions on using base editing and prime editing to fix single nucleotide mutations in therapeutics compared to traditional HDR? Any limitations do you see, or do you believe base or prime editing could be the future direction to explore?
Yeah, I think it's actually exciting that we have different technologies being developed, because I think it will give the opportunity in the future to explore which one might be best for which indication. But in brief, currently HDR can make any single nucleotide mutation. So it is still the broadest. And as I showed, we've demonstrated we can use it ex vivo in basically every cell type, primary human cell type that we can find. Base editing has also been shown to be able to be used both in vivo and ex vivo in primary human cells and in vivo in mouse. But it's limited in that not all changes can be made. And then finally, prime editing is much newer, and so we're waiting to see how well it can work either in vivo or ex vivo. The one advantage of both base editing and prime editing is that they don't require cell division to occur, which may allow them to be used in certain cell types or applications in which HDR is not available because of the requirement of cell division.
Thank you, Matthew. Really cool work, says George. Have you had the chance to do any RNA-seq of these modified LT-HSCs to see if these edits have any global impact on gene expression?
Yeah, that's a great question and something we're very interested in. We have done several years ago a really deep microarray analysis in the immediate aftermath of gene editing and do see a response to the editing process. But we're about to use the increased granularity of single-cell RNA-seq to look at more depth in terms of the function of the LT-HSCs. I will point out it's not simply the editing process that could be perturbing them. It could well be that the cells just being grown outside of the body in artificial conditions is perturbing their transcriptional profile as well. So we're very interested in that.
Great. Another question. When knocking in cDNA into endogenous gene location, how do you limit the expression of the endogenous gene that you targeted?
Yeah. So when we knock into the start codon, we usually include into that cassette a poly A signal sequence. So the transcriptional machinery will read that first knock-in cassette and then terminate transcription at our poly A signal. So the downstream endogenous gene should not be transcribed. If we leave out the poly A signal, it is possible that one could get transcription of both, but we put the poly A signal to stop that from happening.
If you can take this last question as well, how do you limit epigenetic silencing of genes knocked into safe harbor locations?
Yeah, so we try to pick loci that are not prone to epigenetic silencing. We try to make sure we don't include viral promoters and enhancers, which the cell recognizes as foreign and often induces epigenetic silencing. But in the end, we have to safe harbor for its probability of either decreasing or increasing expression in different cell types. And there certainly is some variability. Although I will say it is remarkable that even at loci that are highly lineage specific, like the beta globin locus, we can get ubiquitous expression in a broad number of cell types. So often using a strong promoter, we can overcome any endogenous epigenetic silencing.
Matthew, once again, we'll wrap up now because you have to go. Appreciate your time. Thank you for your presentation, and I hope to see you at the next one.
Thank you for the invitation. Bye-bye.
Bye.